So we are still talking about cellular organelles. These are in eukaryotic cells, and these first sets of organelles that we're going to be talking about are continuous with the nuclear envelope. So the membrane of the nuclear membrane, the envelope, continues into these different things. They're sort of all interconnected. So the first one is the endoplasmic reticulum, and it acts kind of like a biosynthetic factory, and you'll see why that is in just a second. There are two kinds of ER, of endoplasmic reticulum. The smooth ER lacks ribosomes. The rough ER is rough because it's studded with ribosomes. And ribosomes, we talked about, they make proteins. And so rough ER is basically a protein factory. Proteins get made by the ribosomes and fed into this membrane system of the ER. Hey, here's a picture. This shows pretty nicely the way that the ER and the nuclear membrane are connected. See the little tunnels almost out into the ER. And then you can tell the difference between the smooth ER, that's just blue, and the rough ER, which has these little red ribosomes all studded over top of it. You can see that as well here. There is the rough ER with the ribosomes. All those little dots are busy making proteins. And then here's this smooth ER. So both of them are endoplasmic reticulum. Both of them act sort of as factories, but they're going to do slightly different things. The smooth ER, again, you can see that here and here. It is a factory in the sense that it synthesizes lipids. Lipids, remember, are nonpolar. They serve lots of functions, including there are some lipid hormones. So in particular, the testes and the ovaries will have a lot of smooth ER because they need to be able to make testosterone and estradiol. The other thing that smooth ER can do is detoxify poisons. So your liver, for instance, which that's basically its job, is to detoxify poisons of all sorts, things that might harm your cells. The liver would also have a lot of smooth ER so that it can do that function, detoxify those molecules that might otherwise be harmful. There you can see in the liver there's just tons of smooth ER in order to detoxify all of those chemicals that we come in contact with all the time. And by poisons, it doesn't necessarily mean something that would kill you immediately. It's just something that would be harmful to your cells in some way. The rough ER, we're back to our structure determines function. It has ribosomes bound to it. That's its structure. And their function is to produce proteins. So it's really these ribosomes which are producing the proteins. And then they get packaged up in the membranes of the ER and sent off throughout the cell to do various things. So our next is the Golgi apparatus. And so the Golgi apparatus is found sort of outside of the rough ER in particular, but also smooth. And it sort of acts like the post office. It receives vesicles, which are like little bubbles of membrane with stuff inside them. Those bubbles go in to the Golgi where they get modified. If a little piece needs to be cut off, if a little something needs to be added. And then they get sent out to various parts of the cell or of the body. And it kind of looks like, you might have seen this on your coloring as well, it looks like sort of flattened, empty pancakes, or maybe like empty balloons, it's like that haven't been blown up yet. And it's like that because then things can come in, fill it up, get modified, and then be sent off again. This is a really nice picture because it shows the nucleus where the DNA is found. Okay, the nucleus has the code. It sends, when it makes the RNA, it sends the little pieces of RNA out here to the rough ER to produce proteins. And then those little proteins get packaged in these little vesicles. Those are the bubbles that you see there. They float to the Golgi apparatus where they sort of travel through and they get modified. And then they get sent either somewhere else in the cell, like a lysosome might get sent somewhere else in the cell, or if it was something like we were talking about, testosterone and estradiol being made in the smoothie are, that could come to the Golgi, and then that could get sent out to the plasma membrane and then dumped maybe into the bloodstream. So this is just that in words. The code travels from the nucleus to the rough ER. The ribosomes make the protein, the raw protein. The Golgi then modifies the protein. So like I said, if it has to add anything, take away any little piece so it folds correctly, etc. And then those proteins can get sent to the cell membrane for secretion. So insulin would be another example. It gets made in the rough ER in the pancreas and then sent out into the bloodstream to tell your cells to take up that sugar. Our next organelle is the vacuoles. It's really a set of organelles. Vacuoles are those little bubbles that we saw coming off of the Golgi. So they are membrane bound, so they're a little bubble of membrane basically, and they act sort of like storage containers, like Ziploc bags, or maybe a water balloon. In plant cells in particular, the vacuole tends to be much, much larger. You might have seen that on your coloring. And that's where water and starch get stored. So that helps to provide some structure for the plant cell, but also to store food. In animal cells, the vacuoles tend to be much smaller, but there tend to be a lot of them, and they hold all kinds of things. Food, water, waste. They can hold enzymes for various purposes, and then they get sort of tagged and sent to wherever they need to go to do their jobs. So here, this is in a plant cell. It's called the central vacuole, you can see, because it's kind of right there in the middle. There would be the nucleus and the ER and the Golgi. So in a plant cell, the central vacuole really takes up most of the space, and it helps to provide structure by pushing out against the cell wall, which you can see there. It would have lots of starch and water, and so if we were to test a plant cell with iodine, for instance, this part would turn that sort of purplish, blackish color that we saw when we did the McMush lab. This, you can see, is a vacuole filled with lipids, with fats or oils. In this case, the vacuole is so big that the nucleus is kind of squished out to the side, and as is the cytoplasm. So if you think about what kinds of organisms would have lots of these big, huge lipid vacuoles. Okay, here's a couple. In particular, animals that live in the high north or the very, very south, the Arctic or the Antarctic, they're going to have often a lot of fats for insulation. Remember, that was what we said one of the purposes of lipids were. So things like polar bears or seals or whales have a lot of those lipid vacuoles to help insulate their bodies. People as well can sometimes have a similar situation. Okay. Lysosomes are kind of a special kind of vacuole. And lysosomes are the digestive vacuoles. So they are little membrane-bound sacs, again, a little sort of bubble of membrane. And within that membrane, they have hydrolytic enzymes, so basically digestive enzymes. Digestive is what hydrolytic means. It breaks down. So they have hydrolytic enzymes as well as acids. And so when food comes into the cell, or when there's something that needs to be broken down, maybe a cell part that has worn out or hasn't been made correctly, the lysosome will come, and since this is a little bag of membrane and this is a little bag of membrane, they can fuse together because they're all lipids. And so the enzymes can come and break down the food, and then those nutrients can be sent to where they need to go. They can be the source of amino acids for proteins or sugar to make ATP or whatever. Okay, so our next organelles are a little bit different in that they're not part of the same system of membranes that we saw with the ER and the Golgi and the vacuoles. So these are the energy converting organelles. And their purpose, of course, has to do with metabolism, so getting food and turning it into energy. They both have a couple of things in common, which is kind of interesting, and we'll talk about that now a little bit and also later on when we talk about their origin. So first we have the mitochondria, which is sometimes called the powerhouse of the cell. It has some interesting structural pieces. It has a double membrane system, so you can see the outer membrane is kind of like a little capsule, and then the inner membrane is very folded, giving it a lot of surface area, and that's what you see. All those stripes are those infoldings of membranes. The other cool thing that you might notice is that the mitochondria have their own ribosomes and their own little bits of DNA. So that gives us some clues about how they became originally incorporated into eukaryotic cells. The function of a mitochondrion in both a plant and an animal cell, so these are in both, is to break down glucose to produce energy. And the type of energy that our body uses, our energy currency, is called ATP. ATP. And we'll talk much more about ATP later on, but that is the molecule that powers every bit of work that your body needs to do. You eat the food, the glucose, it gets broken down and used to produce ATP, and then that is the fuel. As I mentioned, mitochondria have their own ribosomes and their own DNA, just like a bacterial cell, a prokaryote. And so that suggests that they were once free-living organisms. They were once independent bacteria. And they got eventually incorporated into a larger cell, and then started working with that larger cell. So here's a couple more pictures of the mitochondria. You can see the capsule-like outer membrane, the really folded inner membrane, and then the matrix is where the ATP gets produced in the end. This is just another picture showing the outer membrane and then the inner foldy membrane. And of course, Mr. Wagner, if you've seen him running about, you know that he has lots and lots of mitochondria in his running muscle cells because muscles need to make ATP very quickly so they can keep being powered, keep doing their work. The other type of energy-related organelles are generally, they're called plastids. We're going to focus more on one type of plastid that you maybe have heard of in particular. So the mitochondria are found in all eukaryotic cells, plants, animals, doesn't matter. Plastids are only found in plants and algae, which algae are sort of the ancestors of plants. Does that make sense? They contain special pigments so they can do photosynthesis. So the plastids are where photosynthesis actually happens. And there are two major kinds of plastids. So the one that we usually focus on is called chloroplast. Chloroplast captures light energy and uses it and water and carbon dioxide to make sugars. If you've seen Billy Madison, you will get that joke. Otherwise, you won't. It's okay. So they contain chlorophyll. Chlorophyll are the pigments within the chloroplast, and that's where the photosynthesis is going to happen. So chlorophyll fills up the chloroplasts. That's kind of how I think about it. Similar to a mitochondrion, there are two membranes, an inner and an outer membrane. They're both kind of smooth, but they're both there. There's also chloroplast DNA, and the chloroplast has its own ribosomes, very similar to a mitochondria. Here's just a picture in a plant cell. You can see the cell walls on the kind of rectangular cells, and we'll see some cells like this on the black day. And then all those little green polka dots, those are all the chloroplasts doing their photosynthetic job. Chromoplasts are other kinds of plastids. They contain other pigments. And this is what we see when the leaves change color in the fall. We're seeing a receding of the chloroplasts, and then you're able to see these different colored plastids when normally they would have been sort of overpowered by all the green. And there are a few other organelles, and these are not bound to the membrane. They don't have a membrane. They're sort of a little different. The first of these is the cytoskeleton. Cyto means cell, so we've seen cytoplasm, for instance. Cytoskeleton is a network of fibers, much like your skeleton extends throughout your whole body. The cytoskeleton extends throughout the cytoplasm. It helps support activities and structures in the cell. Things can move along the cytoskeleton. So it's basically the same job that your skeleton would do, just on a much, much smaller scale. Here's a picture with some fluorescent dye, and you can see the green pieces of the cytoskeleton. Another organelle, again, non-membrane bound. This one is only in animal cells, and it is used for dividing the nucleus. So we'll talk about centrioles again when we get to mitosis. But you can see they have a very specific shape. They're kind of star-like almost. And when it's time to divide, they'll travel to opposite ends of the nucleus and pull apart the chromosomes. Here's a picture. You can see sort of right in the middle of all that, and that's where the fibers connect to pull apart the chromosomes. So here you can see the centriole and all of the microtubules extending. The chromosomes would be sort of along this plane, and they would move towards opposite sides of the cell. Couple of more. We're almost done. Next we have cilia and flagella. So these are both locomotor appendages. They're for movement, basically. They're proteins that extend from the cell, and they help the cell to move towards food or towards light or what have you. You can see these are, I think they're trypanosomes, and they have these long flagella, these whip-like tails that sort of whip back and forth and propel the organism along. Sperm cells have a similar structure. Here you have a paramecium, so it's a single-celled protist, and around the outside, it's kind of hard to see in this picture, but you can kind of tell that there's all these little hair-like appendages sticking out, and those are the cilia. And you can kind of think of cilia, if you've seen like a trireme kind of a ship with oars all down the sides. The cilia sort of beat together on opposite sides, and that's kind of how the paramecium moves. If you would like, I can find a little video, and we can look at that, or you can Google paramecium cilia or something and find some cool videos of the cilia there. Here's a cross-section of the cilia. It's got a very specific pattern, and bacterial cilia are different than eukaryotic ones. And it's a big bundle of basically microtubules all packaged together. So, in summary, the cell, it consists of all these different parts, more or less of some depending on the function of the particular cell. But cells really rely on the integration of all those structures to come together and to really do whatever the organism needs to do. So this picture you see at 5 microns of, see the scale there, so 5 microns. The white blood cell is recognizing these bacterium as bad. So it's using some proteins on the outside of its cell membrane to recognize these bacteria. It ingests them by making a little vacuole, pulling them into itself, which we'll talk a little bit more about when we talk about the cell membrane. And then it might destroy them by dumping a bunch of digestive enzymes from a lysosome into the vacuole with the bacteria and then breaking it down. So without all of those pieces working and functioning together, it wouldn't, the whole system wouldn't work. So those are the cell parts. If you have any questions about this, we can certainly talk about it in class on Wednesday or even when we return from Thanksgiving. So, good work.